Welcome back guys. Welcome back to another video session from this channel. So in this video session we will be studying regarding the structural organization of prokaryotic cell and as well as eukaryotic cell in detail. So here if we consider the term prokaryotic it is derived from two Greek words and these include pro and the other one is karyon. So here the meaning of pro is primitive and the meaning of karyon is nucleus. That means all the various living organisms which come under the prokaryotic cells, they possess or they contain only the primitive nucleus. They do not possess or they do not contain well-defined nucleus or complex nucleus. So here if you consider the term prokaryotic, it is derived from two Greek words and these include pro. So here the meaning of pro is primitive and the meaning of karyon is nucleus. So here all the various living organisms which come under the prokaryotic cells, they possess or they contain only the primitive nucleus. They do not possess or they do not contain well-defined nucleus or complex nucleus. So here the various living organisms which come under the prokaryotic cells include bacteria. Is it clear? Bacteria, mycoplasma, pleuropneumonia-like organism and as well as blue-green algae. So the various living organisms include bacteria, mycoplasma, blue-green algae and the other one is pleuropneumonia-like organism. Pleuropneumonia-like organism, which is simply abbreviated as PPLO. Is it clear? So here all these are the various living organisms which come under the prokaryotic cells. So here if we consider the size of the living organisms which come under the prokaryotic cells, so here the size of the living organism is very small. So here once when the size of the living organism, when it remains so smaller, under such condition, the generation time of the particular living organism, it remains so faster. So here all the various living organisms which come under the prokaryotic cells, they are visible only through the microscopic observations. We cannot visualize all these living organisms through our naked eye. So here once when the size of the living organism, when it remains so smaller, under such condition the generation time of the particular living organism, it remains so faster. Is it clear? So here the one of the best example for the prokaryotic cell, here it is nothing but what? Bacteria. We can classify the particular bacteria into different types, so simply basing on the size and shape. So here what are the various types of bacteria which we can classify into different types simply basing on the size and shape? Here the various types of bacteria include bacillus bacteria, which is rod shaped. The various types of bacteria include bacillus, which is rod shape, and the other one is cocci, which is spherical in shape. And the other one is vibrio, which is comma shape. The next one is spirillum, which is spiral shape. So here all these are the various living organisms or all these are the various types of bacteria which we can classify into different types simply basing on the size and shape here. So here though the specific size and the shape of the bacteria, it remains varied, but in case of all the living organisms which come under the prokaryotic cells, the internal organization, it remains very similar. Is it clear? So here all the various living organisms which come under the prokaryotic cells, they possess or they contain the outermost protecting layers like cell wall and as well as cell membrane. So let me write it here. The prokaryotic cells contain cell wall and as well as plasma membrane. Plasma membrane. Is it clear? So here nearly in case of all the living organisms which come under the prokaryotic cells, the cell wall and as well as cell membrane are present, but except mycoplasma. Mycoplasma, it is the only living organism which come under the prokaryotic cells which do not possess these specific protecting layers like cell wall and as well as cell membrane. And in rest of all other living organisms which come under the prokaryotic cells, they possess or they contain the outermost layer as cell wall, and beneath the cell wall cell membrane or plasma membrane is present. Is it clear? And if we consider the structural organization of the prokaryotic cell, the structural organization of the prokaryotic cell, it can be easily understood by taking the advantage of the structure of the bacteria. So here let me show you the structure of the bacteria here. So and if we consider the structural organization of the prokaryotic cell, structural organization of prokaryotic cell. Prokaryotic cell. So here, as I have already mentioned you, all the various living organisms which come under the prokaryotic cells, they possess or they contain cell wall and as well as cell membrane. Except the specific mycoplasma, nearly in case of all the living organisms which come under the prokaryotic cell, they possess or they contain these specific protecting layers like cell wall and as well as cell membrane. And in addition to the cell wall and as well as cell membrane, on the interior side of the prokaryotic cell, here there is an availability of specific fluid filled matrix like substance which is nothing but what? Cytoplasm. So here, in order to understand the structural organization of the prokaryotic cell, so here let me show you the structure of the E. coli. So here these are nothing but what? The protecting layers. The protecting layers of the particular E. coli. So here let me label the things here. Here it is nothing but what? The cell wall. Is it clear? And here it is nothing but the cell membrane or plasma membrane. Cell membrane or plasma membrane. So here on the interior side of the particular prokaryotic cell, here there is an availability of a fluid filled matrix like substance which is nothing but what? Cytoplasm. So here cytoplasm, it is the site where we can observe or where we can visualize certain life activities like protein synthesis during the translation mechanism. And in addition, cytoplasm, it is the site where we can observe the oxidation of the glucose molecule during the glycolysis mechanism. Is it clear? And here on the interior side of the particular prokaryotic cell, here circular chromosomes are present. If we consider in case of eukaryotes, in case of eukaryotic cell, well-defined nucleus or complex nucleus is present. And on the interior side of the particular well-defined nucleus or complex nucleus, here there is an availability of linear chromosome in case of eukaryotic cell. And on the interior side of the particular linear chromosome, the genetic material is packed here. But whereas in case of prokaryotic cells, there is no availability of well-defined nucleus or complex nucleus. On the interior side of the prokaryotic cell, here the circular chromosomes are present here. Is it clear? So let me show you the circular chromosome here. So here it is nothing but what? The circular chromosome. Circular chromosome. And in addition to the circular chromosome, within the interior side of the prokaryotic cell, here there is an availability of certain extra chromosomal circular double-stranded DNA which are nothing but what? Plasmids. So here all these are nothing but what? Plasmids. Plasmids. So here what are plasmids here? Plasmids are the vectors. They play an important role during the gene cloning mechanism or the gene cloning process. They play an important role in organizing the gene transfer. So here, one of the unique characteristic feature of all the particular plasmids is nothing but what? Presence of unique phenotypic character, which is nothing but what? Antibiotic resistance. So let me write it here. Plasmids contain unique phenotypic character. Unique phenotypic character, which is nothing but antibiotic resistance. So here the antibiotic resistance, it is successfully expressed by the means of a specific gene within the specific plasmid here. So here, by taking the advantage of the antibiotic resistance, we are capable to select the transformed cells here. Is it clear? And in addition to this, within the interior side of the particular prokaryotic cell, here there is no availability of any specific subcellular organelles. Is it clear? One of the characteristic feature of the particular prokaryotic cell here, it is nothing but what? Absence of subcellular organelles. Subcellular organelles, they are not observed or they are not present within the cytoplasm of the prokaryotic cell. So let me write it here. No subcellular organelles. No subcellular organelles are present. And the one and only subcellular organelle which is present within the cytoplasm of the prokaryotic cell, here it is nothing but what? Membrane-free ribosomes. So here membrane-free ribosomes, these are the only specific subcellular organelles which are present within the interior side of the prokaryotic cell. And these specific membrane-free ribosomes, they play an important role in organizing the protein synthesis. Is it clear? And here on the interior side of the particular plasma membrane, here there is an availability of smaller infolding like structures which are nothing but what? Mesosomes. So let me write it here. Mesosomes are present on the interior side of plasma membrane. So here presence of mesosomes, it is a unique characteristic feature of all the prokaryotic cells. All the prokaryotic cells, they possess or they contain certain inner folding like structures which remain in association with the surface of the plasma membrane, and these are nothing but what? Mesosomes. So here presence of mesosomes, it is the unique characteristic feature of all the prokaryotic cells. Is it clear? And if we consider the specific cell envelope and as well as its modifications. So here let me write it here. Cell envelope, it is the chemically modified structure. So cell envelope, it is the chemically modified structure which is most commonly present on the peripheral region of the specific prokaryotic cell. So here all the prokaryotic cells, they possess or they contain the specific chemically modified structure which is nothing but what? Cell envelope. So here basically the particular cell envelope, it contains three chemically modified structures. They are nothing but what? Glycocalyx, cell wall and as well as cell membrane. So let me write it on the board. Glycocalyx, cell wall, and the other one is cell membrane, or in other terms it is termed as plasma membrane. So here all these are the chemical modified structures which are a part of the specific cell envelope here. So here each and every layer which is a part of the cell envelope, it is very unique to its structural organization. Though the specific layers, though they are very unique to their structural organization, but all the three layers, they remain in an aggregate form and they act as a single protecting unit. Is it clear? If we consider the particular glycocalyx, it is the outermost peripheral layer, and basically the particular glycocalyx, it contains two different orientations and they are nothing but what? One is the slime layer and the other one is the capsule. So what are those? slime layer and the other one is capsule. So here the specific slime layer, it is so thin, and even the specific loose sheath here. The particular slime layer, it is so thin, and even it is the loose sheath which is a part of the glycocalyx. And in addition to the slime layer, here there is an availability of capsule. So here capsule, it is so thick, and even it is so tough in nature. Is it clear? Slime layer, it is the loose sheath, but whereas capsule is, it is thick and as well as so tough in nature. And here the function of glycocalyx is very simple. The particular glycocalyx, it is responsible for preventing the particular bacterium by undergoing degradation. Because of the presence of the particular glycocalyx, the specific host immune system, it is unable to degrade the particular bacterium. That means once when the particular bacteria, if it enters into a specific eukaryotic organism, the particular eukaryotic host organism, especially in case of animals, they possess or they contain leukocytes and as well as platelets. So here these specific leukocytes and as well as platelets, they are unable to recognize the particular bacterium, or they are unable to degrade the specific bacterium because of the presence of a specific protecting layer which is nothing but what? Glycocalyx. Is it clear? And in addition to the glycocalyx, here there is an availability of one more other protecting layer which is nothing but what? Cell wall. Beneath the glycocalyx, the cell wall is present, and chemically the specific cell wall, it is composed of cellulose and as well as peptidoglycan. So let me write it here. Cell wall is chemically composed of cellulose and as well as peptidoglycan. So here we can classify the specific bacteria into two different types, like gram positive bacteria and as well as gram negative bacteria, simply basing on the chemical composition of the particular cell wall. If the particular cell wall of the particular bacteria, if it has higher concentration of peptidoglycan and low concentration of phospholipid layer, under such condition the specific bacteria, it reacts so vigorously with the gram staining technique, and the specific bacteria here it is termed as what? Gram positive bacteria. In the same way, if the specific bacteria, if it has low concentration of peptidoglycan and high concentration of phospholipid layer on the outermost region, under such condition the specific bacteria, it won't react so vigorously with the specific gram staining technique, and here it is termed as what? Gram negative bacteria. So here the particular cell wall, it is not only responsible for differentiating the bacteria into two different types, like gram positive bacteria and as well as gram negative bacteria, and in addition the particular cell wall, even it is also responsible for providing the size and shape to the particular prokaryotic cell. Because of the presence of the cell wall, each and every prokaryotic cell, it acquires one particular size and as well as one particular shape. Is it clear? And in addition to this, the particular cell wall, it is responsible for preventing the particular bacteria or the particular prokaryotic cells from undergoing bursting or collapsing. Is it clear? And on the interior side of the particular cell wall, here there is an availability of one more other protecting layer which is nothing but what? Cell membrane or plasma membrane. So here the presence of cell membrane or plasma membrane, it is the only common characteristic feature which is observed both in case of prokaryotic cell and as well as eukaryotic cell. Cell membrane is present on the interior side of prokaryotic cell. So here the particular cell membrane or plasma membrane, it is a selectively permeable membrane. That means it won't allow all kind of molecules to come across it. It allows only the certain selected molecules to come across it. So the reason why here it is termed as what? Selectively permeable membrane. Is it clear? And on the interior side of the patellar plasma membrane, here there is an availability of certain infolding like structures which are nothing but what? Mesosomes. So here the specific mesosomes, it can be of three different types and they are nothing but what? Lamellae, vesicles, and the other one is tubules. So let me write it here. Mesosomes are of three different types. Like lamellae, vesicles, and the other one is tubules. These mesosomes, they play an important role in the successful organization of the cell wall. That means for the successful formation of cell wall, these mesosomes, they play an important role. And in addition to this, the patellar mesosomes, even they are responsible for the successful organization of DNA replication. That means for the successful distribution of DNA to the daughter cells, these mesosomes, they play an important role, or these mesosomes are mandatory. And in addition to all these activities, the patellar mesosomes, they play an important role in organizing the secretion process for the successful distribution of the various enzymes and as well as proteins to their destination or to their localization. The patellar vesicles, which are a part of the mesosomes, they play an important role. And in addition to all these aspects, in case of certain cyanobacteria, we can observe one more other modified structure which is nothing but what? Chromatophores. So here chromatophores, these are the specific modified structures which are present in case of cyanobacteria, and these specific chromatophores, they are enriched with certain biological pigments which are responsible for imparting the color. So let me erase the things here. In case of cyanobacteria, cyanobacteria contains modified structure which is nothing but what? Chromatophores, which are enriched with various biological pigments. Is it clear? And here the specific bacteria, it can be either motile or non-motile. Is it clear? So here what is the meaning of motile and as well as non-motile? If the specific bacteria, if it contains certain locomotor organelles like cilia and as well as flagella, under such condition the specific bacteria, here it is termed as what? Motile. And if the specific bacteria or if the specific prokaryotic living organism, if it do not possess or if it do not contain certain locomotor organelles like cilia and flagella, then they are termed as what? Non-motile. Is it clear? So here the patellar cilia, most commonly it is a locomotor organelle, and even the patellar flagella, it is also the locomotor organelle which is present in case of all the prokaryotic cells. Is it clear? So here most commonly the specific motile cilia, they are present on the outermost epithelial surface of oviduct and as well as the respiratory tract. Is it clear? And if we consider the patellar flagella, so here the patellar flagella, it is an lash-like appendage which most commonly protrudes to the outer cell surface of the body. Bacteria can be motile and non-motile. So here simply basing on the presence of the locomotor organelles, we can classify the bacteria into two different types like motile and as well as non-motile. Is it clear? And if we consider the patellar flagella, basically the patellar flagella, it has three differentiated components, and these are nothing but what? Filament, and the other one is hook and basal body. So here all these are the three differentiated components which are a part of the patellar flagella here. Is it clear? So here filament is the longest portion or it is the largest portion of the patellar flagella. Most commonly the patellar filament, it successfully extends from the cell surface to the outer side of the patellar body. Is it clear? So let me arrange the things here. Flagella contains filament, hook and as well as basal body. So here it is nothing but what? The cilia and flagella, right? These are the locomotor organelles. Is it clear? And in addition to this, the particular bacteria, it not only contains certain external appendages like cilia and flagella, even the particular bacteria, it also contains one more other external appendage which is nothing but what? Pillae and as well as fimbriae. So what it is? Pillae and as well as fimbriae are present. So here the particular pillae, it is composed of or it is made up of a specific protein which is nothing but what? Pillin. So here the particular pillae, it plays an important role during the conjugation mechanism for the successful exchange of the gene transfer or for the successful exchange of genes. During the gene transfer, the particular sex pillae, it plays an important role. Is it clear? And if we consider the particular fimbriae, these particular fimbriae, these are the bristle-like structures which are most commonly present on the outermost peripheral region of the bacteria. So here for the successful adherence of the particular bacteria to the host tissue and even for the successful adherence of the particular bacteria to the outer surface of the barren rocks which are present on the surface of river banks, these particular fimbriae, they play an important role. Is it clear? So let me arrange the things here. Then after we will be studying regarding ribosomes and as well as inclusion bodies. Inclusion bodies. So here basically ribosomes, they are ribonucleoproteins. That means they are chemically composed of RNA and protein, and these specific ribosomes, they play an important role in organizing the protein synthesis. Ribosomes are ribonucleoproteins, and most commonly the particular ribosomes, they remain in association with the inner surface of the particular cell membrane or plasma membrane. And here the particular ribosomes, the specific ribosomes which are most commonly present in the cytoplasm of prokaryotic cells, they are nothing but what? 70S ribosomes. And here the specific 70S ribosomes, they are composed of two specific subunits which are nothing but what? 50S subunit and the other one is 30S subunit. Let me write it here. 70S ribosomes are present. And the specific 70S ribosomes, they are composed of two subunits, and these include 50S and the other one is 30S. So here S stands for what? Svedberg unit, which is most commonly obtained during the sedimentation coefficient of centrifugation mechanism or centrifugation process. And here if we consider the specific diameter of the ribosomes, so here most commonly the particular ribosomes, they measure about or they measure around 15 nanometers to 20 nanometers in diameter. So let me write it here. Ribosomes measure around 15 nanometers to 20 nanometers in diameter. And here most commonly the particular ribosomes, they successfully recruit to the particular mRNA during the protein synthesis or during the translation mechanism. So here, once when many number of ribosomes, once when they successfully recruit to the particular mRNA, the specific ribosomes, they are termed as what? Polyribosomes or polysomes. So here, what are polyribosomes and polysomes? Here, polyribosomes or polysomes, they are nothing but these are the group of ribosomes or these are the cluster of ribosomes which successfully recruit to the mRNA during the protein synthesis. So here, let us assume it is the mRNA here. It is the mRNA, right? To the specific mRNA, once when multiple ribosomes, so these are the multiple ribosomes, right? And here the protein synthesis occurs. So it is nothing but what? The protein synthesis, right? So once when the particular ribosomes, once when the group of ribosomes, once when they successfully recruit to the concerned mRNA, now here these specific ribosomes, they are termed as what? Polyribosomes or polysomes. So here polyribosomes or polysomes, they are nothing but these are the group of ribosomes or these are the cluster of ribosomes which successfully recruit to the mRNA during the translation mechanism or during the protein synthesis. Polyribosomes are polysomes are the cluster of ribosomes which recruit to the mRNA. Which recruit to mRNA during the translation. And in addition, within the cytoplasm of the prokaryotic cell, here there is an availability of one more other modified structure which is nothing but what? Inclusion bodies. So here most commonly the particular inclusion bodies, they are responsible for the storage of reserve food materials. Is it clear? And here the particular inclusion bodies, they can be of three different types and these include glycogen granules, phosphate granules and the other one is cyanophycin granules. So let me write it here. Inclusion bodies responsible for the storage of food material. And here the particular inclusion bodies, they can be of three different types and these include glycogen granules, phosphate granules, and the other one is cyanophycin granules. And in addition to this, in case of certain other bacteria, some more other specific inclusion bodies are present and these are nothing but what? Gas vacuoles. So here gas vacuoles most commonly they are observed in case of green surface bacteria and as well as purple bacteria. Is it clear? In case of certain bacteria, gas vacuoles are present. Gas vacuoles are the inclusion bodies. Inclusion bodies in case of green sulfur bacteria, purple bacteria. And even in case of the photosynthetic bacteria. Thereafter we will be studying regarding the structural organization of eukaryotic cell. So here what is eukaryotic cell and what are the various structural components which are a part of the eukaryotic cell in detail. So let me arrange the things first. And if we consider the structural organization of eukaryotic cell, basically the term eukaryotic, it is derived from two Greek words, and these include eu and the other one is karyon. So here the meaning of eu is good or well defined, and the meaning of karyon is nucleus. So let me write it here. Eukaryotic. So here the meaning of eu is good or well defined, and the meaning of karyon is nucleus. That means the specific living organisms which come under the eukaryotic cells, they has or they possess well defined nucleus or complex nucleus. So here what are the various living organisms which come under the eukaryotic cells? Here the various living organisms which come under the eukaryotic cells include plants, animals, fungi and as well as protists. So let me write it here. Plants, animals, fungi and as well as protists. So here all these are the various living organisms which come under the eukaryotic cells here. Is it clear? On the interior side of the eukaryotic cell, there is an availability of well-defined nucleus, a round spherical ball-like structure which is nothing but what? Nucleus. So here the particular nucleus, it is responsible for controlling and coordinating the life activities of the eukaryotic cell. So here it is nothing but what? The nucleus. So here the particular nucleus, it is responsible for controlling and coordinating the life activities of the eukaryotic cell. In case of prokaryotic cell, there is no availability of well-defined nucleus, but whereas in case of eukaryotic cell, there is an availability of well-defined nucleus or complex nucleus. And on the interior side of the eukaryotic cell, cytoplasm is present. On the interior side of the eukaryotic cell, a fluid-filled matrix-like substance, which is nothing but what? Cytoplasm is present. So here in case of the eukaryotic cell, the particular cytoplasm, it undergoes, or the particular cytoplasm, it has extensive compartmentalization of various subcellular organelles. In case of the prokaryotic cell, the particular cytoplasm, it only contains membrane-free ribosomes. But whereas in case of eukaryotic cell, if we consider the particular eukaryotic cell, within the cytoplasm of the eukaryotic cell, here there is an availability of many number of subcellular organelles. Is it clear? Cytoplasm of eukaryotic cell has many number of subcellular organelles. So what are the various subcellular organelles here? These subcellular organelles include endoplasmic reticulum. So here it is nothing but what? Endoplasmic reticulum. On the peripheral region of the endoplasmic reticulum, ribosomes are present. So here due to the presence of ribosomes, so here these are nothing but what? The ribosomes. Due to the presence of ribosomes on the peripheral region of the endoplasmic reticulum, it induces a rough appearance or it induces a zigzag appearance. So the reason why the specific endoplasmic reticulum, here it is termed as what? Rough endoplasmic reticulum. And in addition to the rough endoplasmic reticulum, even the smooth endoplasmic reticulum, it is also present. So here it is termed as smooth endoplasmic reticulum because of the absence of ribosomes on the peripheral region. In addition to the endoplasmic reticulum, here there is an availability of some more other subcellular organelles like Golgi apparatus, mitochondria, lysosomes, peroxisomes, and even in case of plants, there is an availability of some more other subcellular organelles, and these include plastids and as well as vacuoles. Is it clear? Though the specific plants and as well as animals, they come under the eukaryotic cells, but there are certain exceptions in between the plants and as well as animals. In case of plants, plastids are present and as well as vacuoles are present, but whereas in case of animals, plastids and as well as vacuoles are absent. Is it clear? And in addition to this, in case of animals, centrosomes are present, but whereas in case of plants, centrosomes are absent. So let me write it here. So in case of plants, vacuoles are present. Vacuoles and as well as plastids are present. But whereas in case of animals, these are absent. Is it clear? And in addition to this, in case of plant cell, the outermost protecting layer is nothing but what? Cell wall. But whereas in case of animal cell wall is absent. Is it clear? Animal cells, they acquire or they possess flexibility because of the absence of cell wall. But whereas plant cells, they are so rigid, they are always so erect because of the presence of cell wall. Is it clear? And in addition to this, there is one more other exception in between the plant cell and as well as animal cell, and here it is nothing but what? Presence of centrosome. In case of animal cells, centrosomes are present, but whereas in case of plant cells, centrosomes are absent. So let me write it here. In case of animal cells, centrosomes are present. But whereas in case of plant cells, centrosomes are absent. So here these particular centrosomes, they contain specific barrel shaped structures which are nothing but what? Centrioles. And these specific centrosomes, they play an important role in aiding the cell division mechanism in case of animal cells. Is it clear? So here this is regarding the structural organization of prokaryotic cell and as well as eukaryotic cell in detail. So here in the next upcoming video session, we will be studying regarding the structure of cell membrane and as well as plasma membrane in detail. So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends. And I remember you people to subscribe my channel for getting more and more videos what I make day to day. Thank you.